is called The Neuroscience of Misophonia. So brain research and possible neuro treatments. That's what I'm going to talk to you about today. And let me give you a brief outline of the structure of my talk. So the first part of my talk, the first third, will consist of an introduction to the human brain. I'll talk to you a little bit about the structure, some of the operational principles and function of the human brain from the way I studied the brain during my PhD work and some of my research thereafter and at UM. I'll give you the two sort of classic studies now, evidence that misophonia has neurological conditions, neurological causes, I should say, or correlations, because that's really what neuroscience is. You're looking at when things happen in the brain correlated to outside stimulus. And so there's really a correlation between the outside stimulus and what goes on in the brain. We can't really say, sometimes we can, whether those stimuli cause these changes in the brain. So I just want to make that distinction. The first study, The Brain Basis of Misophonia, came out with Dr. Kumar et al. In 2015, I think, and I was actually a subject in that study. It was held in London. Could tell you about the personal experiences if you want to ask me during the Q&A, that's fine. And then a second fMRI study, this was an fMRI study, a second fMRI study came out essentially reproducing, in some ways replicating, which is really excellent, this work. So this is really a crucial point, to have reproducible results in a disorder so young in terms of being studied scientifically is amazing in my view, in my opinion. And that's really a very kind of hopeful thing. And then the last thing I'll tell you about is, as I said, two possible neuroscientific treatments. This is what my grant is focused on in which we're waiting to hear back soon, and maybe in 6 weeks or 2 months or so, whether the grant went through. Okay, so let's begin with a poll. I'm going to teach you a little bit about the brain first. Okay, let's begin with a little bit of a poll. I just want to sort of gauge your knowledge here about the brain. Okay, I do this all the time to talks that I give similar to this. So how about this? True or false? We only use 10% of our brain's capacity. We only use 10% of our brain's capacity. So what I'd like you to do without doing an official poll is just go in the chat and type true or false, okay? Go in the chat and type true or false. And I'm putting it on the screen here, okay? So everybody can see. Excellent, this is great. This is great, everybody, thank you. We have how many people on the call? 32, so I don't have an exact number, but I'm going to scan this and you can see it as well. Actually, maybe I shouldn't put this on the screen so everybody sees everybody's answers. I can't, I have to. Okay, so true or false? We only use 10% of our brain's capacity. Only use 10% of our brain's capacity. So like true, 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 1 false, 2 false, 3 false, mostly true, and a lot of falses here. Okay, I would say probably I'm just going to estimate maybe like 65, 70% said true. Okay, if everybody is acceptable to that. Okay, so that's the first, what's called neuro myth. This, folks, is false. Okay, this, folks, is false. And let me put this here. All right. Let me explain why really briefly. So advertisers, psychologists, parapsychologists, self-help techs, life coaches, a lot of other people love to use this myth. There's a whole bunch of these pop neuroscience, if I could use that term, which talk about these different brain facts. And so this is actually false. We almost use all of our brains almost all of the time. We can imagine the functioning of the real, we can now image, the functioning brain in real time, and there's never any inactive areas of in healthy brains with respect to the task. So I'm going to talk in a little bit later about these, what are called brain networks. So thanks to this neural imaging technology, we can now image the brain at rest, which I'm going to talk about as well. That's a very important aspect, which is involved in misophonia, the brain at rest. And also the brain doing a task, like you're all paying attention to me now. And just consider how the brain uses energy. It would be actually metabolically impossible if the brain used all of this, almost all of the time, energy to balance out information processes and energy consumption. So just from a metabolic resources, it wouldn't even be possible to use just 10% of the brain. And there's a state of there's a cognitive state that's being well studied now. And I mentioned I was a peak performance with the Flow Research Collective. There's something called flow. Flow is when you're in the zone. It happens to athletes, it happens to painters. It was well studied by, I'll make a first book recommendation here, by a psychologist named Mihai Csikszentmihalyi back in the 60s, and he wrote a book called Flow. Flow is defined as an optimal state of consciousness where we feel our best and perform our best. When you lose your sense of self, you lose your sense of time, you become really proficient and a flowy kind of when you're performing these skills or a task, like in sports or really in anything. Jazz musicians call it in the pocket when they're improvising, runner's high, these kinds of things. And you would think that when you were super focused, we're using a lot more of our brain. And actually, we found out that we use less of our brains when we're in a state of flow. And flow, and neurobiologically, may actually be the opposite of chronic stress or something like PTSD. The same neural mechanisms may be involved, but divergent pathways. Okay, guys, ladies and gentlemen, the next true or false. I can't lose my train of thought. So let's do in the chat again, okay, folks? Some of us are left-brained or right-brained due to what's called hemispheric dominance, and this helps explain differences in how we learn. So people, you've heard left brains are more analytical and logical, and right brains are more, creative, have a higher imagination. So let's go in the chat again, if you will, and type true or false. People are left-brained or right-brained, more left-brained or right-brained. Let's bring up the chat and let's look. Okay. Great to see. Great to see. True, false, true, false. True, false. We're looking. False is the newer thinking. Susan, nice to see you again. Nice to hear from you again. Equal. Nope, you can only get one. True or false, Martha? Looks like about 50-50. Let's say everybody answered, just in lieu of time. So it looks to be about 50-50. This is false. Okay, this is false, folks. There's no really such thing at the level of the brain in terms of being a left brain person or a right brain person. Let me just give you a few examples. There is something called functional lateralization. Structurally, almost every structure that exists in the left brain exists in the right brain. But functionally, there is some lateralization. Research has shown that math skills are used by both sides. Creativity actually uses both sides. A study just came out literally 3 weeks ago or a month ago by John Cunios, one of the world's experts on the neurobiology of creativity out of Drexel University in Philadelphia. He found that he was looking at jazz improvisation and creativity of jazz musicians. And he found that actually that novice creativity is recruited by mostly the right side of the brain, but expert creativity, so experts in jazz improvisation, was much more localized to the left side. So creativity, depending on whether you're a novice or expert, depends on this left brain or right brain. But as a personality trait, as a character trait, there's no real such thing as being more left brained or right brained. Okay. And a study just came out of 1,000 subjects. That's huge, out of Utah. This is a few years ago, actually now, 2 or 3 years ago, I think. They said it's absolutely true that some brain functions occur in one brain or the other side of the brain. Language tends to be on the left and attention tends to be on the right. That's functional lateralization. But people don't tend to have a stronger left or right side brain network. It seems to be determined more by connection. Okay, so don't think about yourself as being left brain or right brain. All right, so those are two neuromyths that have been really well debunked now. Okay, that's it for the polls. There's a bunch of other neuromyths as well. For example, multitasking. There's no such thing at the level of the brain as multitasking. The brain really only pays attention to one, maybe two things. What people do is actually switch their attention between different things, and the metabolic resources then increase and cognitive load increases. Cognitive load is the amount of information processing at one point of the brain. And what we want to do is lower cognitive load so we can better focus on tasks and things like that. That helps with, for example, misophonia, in my opinion. All right. Okay. Well, let me skip this slide. So just in the lieu of time. Okay. All right. So Jen, you could just feel free to stop me if you want, or slow me down. Okay. So I'm going to teach you a little bit about the brain. Okay.
You're
Awesome.
doing fine, Michael. I think this is great. Just we're okay
Cool.
with time.
Okay, good. So folks, I have my PhD in complex systems and brain sciences. So I study the brain from a complex systems point of view, and that's really how we need to look at the brain, especially to cure things like Parkinson's, schizophrenia, autism, and misophonia. Um, it's going to come from looking at the brain as a complex adaptive system. Let me explain what that means so you have more context, okay? Um, so this is, I love this. I've shown this before. This is out of Adam Ghazali's lab at UCSF. This is called the glass brain. You can just look it up on YouTube. This is his brain, actually. This is a computational visual model of a brain playing a video game in real time in virtual reality. And you could see how beautiful it is. The electrical signals traveling to and fro from different areas of the brain in these large scale brain networks. You can see different areas of the brain lighting up, like red, green, blue. Those are the different brain waves associated with different cognitive functions. And they're dancing around. The way I like to describe it is it's like called a glass brain. You can fly through it. You can see the different connections. These are real signals traveling in real time. You can see this is the corpus callosum connecting to two areas of the brain. And these different brain areas are coming together in sync and connecting. And at the same time, believe it or not, they're disengaging really super rapidly. And the metaphor that my advisor likes to use, Scott Kelso, is you can think of it like a good old-fashioned square dance. People are coming together, dancing, then leaving each other, and then dancing with somebody else, and then leaving and dancing with somebody else. That's how these brain areas are connecting. And it's this brain connectivity that's really important. Let me give you some other examples of complex systems. So let's start off with the ants. So you can see these ants. They're creating a bridge. This is a spatial and geometrical pattern. It's a functional pattern out of the structure of the ants. No one ant has a concept of a bridge, right? They're not civil engineers, but each ant is following local rules, and they evolve to create a bridge when they need it. It's a beautiful, amazing biological system. The same thing with these starlings. This is called murmuration. You can see, hold on, let me mute myself. I gotta clear my throat. So you could see these beautiful starlings are creating these beautiful spatial patterns. And they have evolutionary, they serve evolutionary purposes as well. They can send signals of danger from one end to the other, a huge wave that goes through the entire system, because each bird, each starling is following local rules. And like, don't fly so close to me, I won't fly so close to you, let's maintain sort of same speed as the bird next to me. And out of those local rules, global emergent, the word here, patterns arise. This is what's called self-organization. There's no top-down organization by design. There's no one bird, like a conductor, like, everybody plays this set of instruments, everybody plays that set of instruments, and together I'm conducting and leading the whole thing. That's top-down organization by design. That's not how biology works. Biology works, organization is from the bottom up. It's self-organized. The same thing really happens in the wave, as I already played the video for you. I already gave it away. The wave in a sports arena occurs very similarly. It's a self-organized pattern, and hurricanes are very structured, organized, spatial weather patterns that each atom and molecule, water molecule, is following local rules based on the system. So that's how the brain works. I just wanted to give you an idea of how the brain works, okay? Let me skip this slide in lieu of time. I'll come back to that if you all want me to in the question and answer session. But I just wanted to give you some idea of fascinating brain experiments that are being done right now. Maybe I'll come back to it if I have time. So that's sort of brain function. Let me talk to you a little bit about brain structure. So here's a picture of a brain. This is actually my brain. This is actually, by the way, yeah, this is the MRI shot I got when I did Dr. Kumar's study. So he gave me my data. So it's cool. I have my own brain and how it lights up. And I always make jokes. You can see all the holes there, like everywhere. No, I'm just kidding.
Michael?
Yes, go ahead.
I don't know if is this, I guess, his first paper you're talking about, right?
This is Dr. Kumar's The Brain Basis of Misophonia that was published in Cell Biology.
Okay, if there's anything, I don't know, but if there's anything that you can show us on here, please do.
On this study?
Yeah, I mean, in other words, while we're seeing this, I mean, maybe we'll probably talk about that study later, but it'd be interesting to see on your own brain what the results of
Oh,
the studies are.
oh, well, I do have, I'm going to go into this study in depth a little bit later in the
Okay.
presentation. I have a set of slides, but if you want me to talk about it now, well, you see, this is the thing though. So you can't, there is, look, I'll show you. Um,
I'm sorry, I didn't mean to throw you off. I just thought it would be
no,
so.
no, 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 you didn't throw me off. It's okay. It's okay. Let me show you here. So look, this is the study, the brain base of misophonia. Okay. There's the abstract. And let me just show you. So look here. So you could see this area of the brain lighting up. That's the right anterior. That's the anterior insula, which has been shown to be really involved in misophonia. I'll go into this study a little bit when I get to this slide. But I just want to tell you that he recruited, I forget however many people in the study, 20 some people with misophonia, 25 people and 20 controls. You cannot, with neuroimaging currently, with fMRI studies, look at the brain areas lighting up for a single individual. It's statistically averaged, okay? So when you see this, this is not one brain. This is like a statistical average of 20, 25 brains. And this area lighting up is the statistical average of all of those light activations in the brain. Does that make sense? Okay, yeah. You want me to talk, so I'll come back to it. I'll come back to it. Okay. Now, if I'm going too long, just let me know, and I'll come back to it. But yeah. So my point is in this is my brain. This is structural fMRI. This is structural MRI. So with structural MRI, you can get a picture of your brain, right? But in functional MRI, fMRI, that's the brain areas lighting up due to certain disorders or whatever. In that, you can't get a single picture. You have to have like a lot of people. You can with one person, but it won't give you very reliable data. That's why you need to bring in statistics. And so yeah, this is my brain. It's the most complex system of which we know. And there's about 86 billion neurons and many more glial cells and about 100 trillion connections. So each of the neurons is connected to about 5,000 or 10,000 other neurons, totaling about 100 trillion connections. So, more connections than there are galaxies in the universe or stars in the Milky Way galaxy. So massively, massively complex. Okay, and you can see the different gray matter and white matter of the brain. And I'll explain what that means in a couple slides. But you can actually see it right here, like the gray matter is the outer part, and the white matter are these long connections, these axons that connect different areas of the brains. And that's really, that's the most important part in my view, is the brain connectivity, okay? So normally we would parcellate the brain into different lobes, the visual cortex in the back, the occipital lobe, the parietal lobes, which are involved in sensory information, the frontal lobes, and then the auditory cortex, the temporal lobes over here. We now know that the brain can actually be parcellated into hundreds of different individual areas. And it's these areas that are coming together and dancing around in these different ways that I was talking about when I showed you the glass brain before, okay? So this is actually back to Jen's question, which was relevant. This is the neural activity measuring methods. So fMRI was really good at spatial resolutions. So it's really good at localizing exactly where in the brain something happens when you're, for example, hearing a trigger sound. But it's really bad at time resolution. And that's one of the reasons why you can't actually just look at one brain. Time resolution, meaning when did this area of the brain light up versus the person hearing the trigger, when the trigger sound was played, okay? On the other hand, EEG, this person's wearing an EEG cap, this is the opposite. So this is bad, really bad at localizing where something in the brain happens, but it's really, really good at localizing when that thing happens versus when the person hears a trigger sound. Both methods are being used to study misophonia, okay? And you need both methods. So this is just a picture of an axon, the heart of the brain, the essence of thought and action, This is the functional unit of the brain, if you will. Signals come in from these dendrites, and then they culminate together, and then they send a signal, an electrical signal, down the axonal path. That's the white matter, to these synapses, which I'll talk to you about in 1 second. And then that translates to neurotransmitters, which then signal the next neuron. Now, this is really like an action potential. I think I actually put oh no, I didn't put a picture of one. But an action potential, to think about it, is really like lightning. That's really almost not even analogous. It's almost literally how the brain works. Like a cumulonimbus cloud bubbles up and then separates the charges from the positive up here and the negative down here. And then when that charge builds up, you get an electric spike. It's almost the same of how electric spikes travel down these axons. So everything about you, your thoughts, your desires, your wishes, your misophonia, if you have it, your love, your desire, your anger, all of these things that make up who you are, these emotional states, everything about you, your sensations of seeing color, all of that really boils down to the best we know about neuroscience is these electrical signals traveling down axons. And here's just a picture of the enormous complexity. Again, this is like 1 millimeter of human brain tissue. You could see the massive complexity here, okay? I will highly recommend, highly recommend, I put this in every presentation I give, these 2 books by Joe Ledoux. Oh my God, this is great though. I didn't even realize it, that Jen, that I have this in all my presentations. The talk that I just sent you beforehand, I put this in that presentation. So I will highly recommend this book, Joe Ledoux's book called The Synaptic Self, How Our Brains Make Up Who We Are. Please read this book. And Anxious is also a great book. I love Joe Ledoux's work on anxiety and fear. He's pretty much like the godfather on the amygdala and fear and anxiety in the brain. Shortlist for the Nobel Prize, I think. This guy's one of my favorites. So I always put that in there. The reason why is because the synaptic self is really, again, who you are. All of these signals traveling down and how these synapses are functioning, and how the synapses are changing. How am I doing on time? Everything is good?
You are doing fine on time. I know you probably feel rushed, but it's so fascinating. So
okay,
just do your best.
Okay, cool. If you want to stop me for questions, I can do that. Maybe take 1 or 2 questions during. If you want to do that's fine, or we could just wait till after. So
Far, we're not getting any questions. And just to remind everybody, if you want to ask questions during the presentation, I will do my best to answer them, and then we'll go back to Dr. Mannino at the end. So
cool.
I see questions
Great.
coming in now. So just keep going, and I will do my best to answer.
Okay, cool, awesome. So I just wanted to tell you about a couple of brain areas that I'm really interested in that are largely due because of misophonia and that were found to be involved in misophonia in these studies. And so the hippocampus is one. So folks, the hippocampus is the part of the brain involved in learning and memory. It's involved in other things too, like spatial awareness, okay? And it's like sort of the brain's GPS system. And I'll show you a picture of my, the hippocampus is it located there? Yeah, it's right there deep inside the temporal lobe of the brain. And you could see my hippocampus right here. This is my hippocampus. And these are my hippocampi, okay? I have a hypothesis, and this was a little bit more than speculation now, that, the acquisition of new triggers are learned. And so Dr. Kumar and colleagues found that the hippocampus is highly involved in misophonia, in these large-scale networks that I'm going to talk to you about in a second. And so it could be, this is one of the reasons why you have an abnormal connectivity with the hippocampus and other areas of the brain that are involved in the acquisition of new triggers. And so it's the certain disorders that it's involved in: anterior great amnesia, famous Alzheimer's disorientation. And so just a very famous study on the synaptic self and on neuroplasticity, for example. Neuroplasticity is the brain's ability to rewire itself due to incoming stimulus. It's a tricky word. We can talk about it in the Q&A session. But I just wanted to, this is one really good example of the brain changing its function, changing its structure based on this localization of function of incoming stimuli. So this London taxi driver study is very well known now. The London taxi drivers have to take this massive test called the knowledge. It's called the knowledge, and they have to study for years, and you can only get a few times to take it because you have to master the routes in London, which are not, they're kind of self-organized if you think about it. They're not like grid-like. They're all crazy if you've seen a map of London. And so they did a study on the hippocampi of the London taxi drivers and found famously that London taxi drivers have an enlarged gray matter. And the gray matter, by the way, I'll just go back here. I just forgot this in a second here. But so the gray matter, okay, is the gray matter. I'm pointing to my screen. You can't see. The gray matter here is the cell bodies. The white matter is these axons. I forgot to mention that. Okay. They found that the hippocampi were enlarged in London taxi drivers versus normal controls. Really fascinating. Really fascinating. The amygdala. I'll talk a lot about the amygdala. It's very involved in stress and anxiety and emotions like fear and sadness, and the fight, flight, or freeze response as well, and the associated disorders, lack of empathy, depression, fear recognition, aggression, irritability, PTSD, things like this. And Joe Ledoux wrote this book called Anxious, and where he kind of reignites and he sort of overturns a problem that he actually helped promulgate actually from William James, called the James Lange theory of emotion. And it's really phenomenal. I just love to tell you about it. I don't want to tell you about it now. If I have time, we can come back in the Q&A session. But it's really phenomenal, and it involves the amygdala, not as a fear center of the brain, but mostly as a threat detection center of the brain. And that's different than the emotion and the feeling of fear. Okay, he separated these two things out and overturned a lot of work. It's just really incredible. We can get into that a little bit later. And so, yeah, one of the, I'll just mention this now actually. So one of the things that he found, and that other people found as well, is what he called the low and high roads to fear. So here's the amygdala, here's the sensory cortex, which is this, if you let me use my brain model here, folks. So here's the brain. The sensory cortex is what's referring to this outer layer, right, that I showed you. If you took this cortex off, it would be like a small pizza and put it out. This is the neocortex of the brain, the last part of the brain to evolve. And the amygdala is deep inside the brain. So if I break the brain up here to two halves, I do what's called a hemispherectomy. The amygdala is right there, deep inside the brain, near the hippocampus actually. And so this part of the brain, right deep in the center too, is called the thalamus, sorry. And the thalamus is like the relay station of the brain. All of the sensory information from the outside world comes in and goes to the thalamus first, and then gets relayed out to the different parts of the sensory cortex. But there's a path. So if an emotional stimulus comes in, okay, and it goes to the sensory thalamus first, it forks off into two roads. It goes to the quick and dirty road to the amygdala really quickly and bypasses going to the sensory cortex. Then there's also the long road where these signals go to the sensory cortex and then go to the amygdala. And then you have the emotional response, right? Like your pupils dilate, you start sweating, you have the fight, flight, or response, the sympathetic response. And this is really important. This is actually very important in Joe Ledoux's theory of emotion that he wrote about in anxiety. And so I just wanted to point that out to you. And I'll tell you about more of the importance in a couple of slides when I come back to this. But this is just an idea. Okay. The insula is another area of the brain that's very much involved in misophonia, and we'll come back to this when we talk about Dr. Kumar's study. So the insula is right here. Okay. You have to peel back sort of a part of the brain, and it looks deep inside the brain. And I'll show you a picture of my insula. You can see my insula is right there. Okay, the insula is one of my favorite parts of the brain, if not my favorite part of the brain. It's really involved in things like body awareness, interoception. So if you can feel your own heartbeat, the amygdala is active. And interoception is very, very important for misophonia. And I'll tell you why, because Dr. Kumar's study found that misophonics or misophones have higher levels of interoception. And this is connected to Joe Ledoux's theory of stress and anxiety. And I'll explain why.
And Michael, if I
Yeah,
could interrupt and add just a little bit about
yeah,
interoception, just to make it a little clearer,
yeah, of course.
interoception is another sense, like you have hearing,
Right.
touch, seeing. It's a sense, though, that's not often talked about. And as Dr. Mannino said, the sense of interoception that you have kind of dictates how much of your internal body functioning you actually are aware of and feel. So some
Exactly.
people feel a much higher level of what's going on in their body, and some people feel less.
Yes, and so interoception is really I love the concept. If you want, you can actually go to, I could put it in the chat here. You can go to the multi-dimensional assessment of interoceptive awareness. Sorry for the misspelling. I'm typing fast here. And you could take that that questionnaire to measure your levels of interoceptive awareness. Now, let me tell you something really interesting. So there's one of my other favorite neuroscientists is a guy named Antonio Damasio. And Antonio Damasio wrote a book called Descartes' Error. And what he found, something very interesting, is that people's level of interoceptive awareness affects their decision-making processes. So he found that people who are more sensitive to their own heartbeat, for example, are better at performing cost-benefit analyses in this task that he designed in his lab called the Iowa gambling task. So there's a very deep connection, and Joe Ledoux talks about this as well, between your body and the body awareness and other cognitive processes, okay? And again, people with misophonia, the anterior insula is like the part of the brain that was really found to be involved in misophonia, and we'll talk about that in a second. Okay, moving along quickly. And so I just wanted to bring your attention. This is a picture of the structure of the brain, these axons, and the brain really works in what's called large-scale brain networks. That's what I studied, a part of the brain, these networks that are involved in different cognitive functions. So we used to think, right, that this area of the brain is involved in this area of the brain is involved in that, this area of the brain is involved in this other function, and we now know that that's not true. We now know that cognitive functions and pathologies too are not individual areas. They're individual areas connected to one another in that dancing that I was talking about before in the large scale brain networks. And they're called small world networks, actually. So if you know what small world networks are, if you've heard of that term, these are the kinds of networks that are involved in the brain and involved in misophonia. And so one of the most famous networks is called the default mode network. And this is a very important network. It's the network of the brain that is involved in misophonia as well. So let me see how to talk. So this is the brain's default mode. Okay, what they did was Marcus Rako about 15 or 20 years ago put people in those fMRI scanners that I was talking about earlier and said, don't do anything, just whatever, ruminate, daydream, think about yourself. Really, you're not involved in any specific task like listening to trigger sounds or a finger tapping task or watching a movie or paying attention to something. And they found that when the person's really not doing anything, there was a very highly structured and organized network of the brain that springs into being. And it turns out that this network is called the default mode network. It's when you're not really doing anything, when you're just thinking about yourself or the future or just daydreaming or mind wandering. And they found that this network is really, really involved in a lot of different important things in human life. So they found that meditation leads to reduced default mode network activity. And this is a good thing because when you have too much default mode network activity, you're too focused on yourself. And that is connected to autism. It's connected to depression. So increased default mode network connectivity in individuals at high risk for depression. So you can't too much focus on the external world and the task at hand. You're too much focused on mind wandering and inside yourself. And so meditation turns out to reduce some of this network activity. It's been an exercise has an effect on this network as well. It's been involved in the social understanding of others. So that's, again, back to autism. So the connections between the different nodes in the default mode network are involved in understanding other people. Like, for example, I know that even though I can't see you all now, I know that you're paying attention. I know that you're interested. I know that you're learning. And that area of the brain and the connection is lighting up in the default mode network. Okay, if that sort of makes sense. So I know that other people have mental states and desires and wishes and are listening to me. And that's really important. So when this abnormal functional connectivity happens in the default mode network, it leads to problems with social interaction and social communication. And even decision making, like moral decision making, like doing the right thing and empathizing with other people. Okay. So it's really important, the network. It's involved in stress as well. So there's other networks of the brain too, as well. And I just want to skip that and go to this slide here, just in the lieu of time. So this is a very important slide here because there's other networks of the brain too. One of those networks is called the salience network, and another one of those networks is called the central executive network. And so the central executive network is involved in this decision-making processes when you're actually involved in a task. And the salience network is involved in what stimuli are salient, either externally in the external world or internally from your body. And it's the switching between these networks, the balance between these networks, coming active, going down, coming active, going down, that really serves for proper mental functioning and cognitive functioning. And that's really like one of the takeaways here about these different networks. The salience network is really involved in misophonia, and that's what Dr. Kumar found, as I'll talk to you about in a second. Both the salient and the default mode network. Okay. All right. And so it's the ability to switch. And okay, so let me, actually, I could do this here just to if nobody's seen this before, I'll do this here. Talking about selective attention and being able to pay attention to things and not being able to multitask. This is famously called the inattentional blindness experiment. And so I'm going to play this video, and hopefully you'll be able to see it and you'll be able to actually do the video. So this is not in a group setting, this is individually, but try your best to actually do this exercise in the video. Okay, so I'll play it now. All right, here we go. Oh, and you won't be able to hear because of this. Hold on, let me see it. Hold on. Well, you're not going to be able to hear, but it's fine. Okay, so this is an awareness. So you're supposed to count how many passes does the team in white make. Okay, so try that. Focus, pay attention, and count how many passes the team in white makes. Okay, here we go. Ready? Go. How many did you get? How many did you get, folks? You can put it in the chat. 13, 10, 13, 13, 13, 12, 9, 3. Martha, you meant 13, I think, right? Okay. 12, 11, 13. Good, good. Excellent, excellent. Let me go back to it. Let me go back to it here. Let me go back to it. Okay. So the answer is 13. You're correct. Okay. The question is, did you see the moon walking bear? Ah, shoot. Did everybody see the moon walk? Can you still see my screen, Jen? Okay.
Absolutely, yes.
Okay, here we go.
And I do see the question, did you see the moonwalking bear?
Yeah.
Which I did not see before.
Sorry, I just gulped it for everybody. Sorry. Okay, so it's easy to miss something in knowledge. It's a famous test on inattention. This is called inattentional blindness. And so I have a theory. This happens with audition too, as well, right? This is called the cocktail party effect, or something called sensory gating. Incoming information gets gated and filtered out. When you're in a cocktail party and you're talking to somebody, you're focused on one person, and you ignore information. And that's one of the the top job of the brain is actually to filter out incoming information. And I have a theory that we could actually test, hypothesis that you could actually test. So would there be a difference in misophones versus controls on things like intentional blindness, or what's called change blindness, for both vision and audition? So I think that would be something very interesting to test for, and see if there's a difference, because sensory gating is a problem, right? I have a huge problem with sensory gating, being a misophone myself. And an interesting study too, Jen, I think you and I talked about this before, found that there's actually increased levels of creativity with people who have problems with sensory gating. And this makes sense because different brain areas which are abnormally connected have a problem, right, with misophonia, but they may have sort of like a silver lining too, instead of creativity. So that's just an interesting hypothesis. Okay. Okay, so let's talk about these two studies. All right. So this is the first study, the brain basis for misophonia, Dr. Kumar's study. What did they find in this study? What did they find in this study? So let me talk about the first thing they found in this study is they used galvanic skin response. So they actually put electrodes on the fingers to sense because when you're aroused, right, by an emotional stimulus, the conductivity of your skin changes because the threat detection circuitry, the amygdala, the output of the amygdala goes to the hypothalamus and other areas that ignite the fight or response. The conductivity changes, so you can measure that electrically when you are aroused. And so it was the first like sort of objective metric over and above like just subjective reports and qualitative reports that yes, I get triggered by sounds. No, we now know that you are getting aroused because your galvanic skin response is shooting off when you're hearing these trigger sounds versus when you're not hearing these trigger sounds. So that was like sort of the first objective measurement, and Mirren Edelstein et al. Did that. They also replicated that here. So what they found in this study is very interesting. I'll go through these two studies right now. So yeah, you could see that they first did the rating. The trigger sounds in the misophonia were bad. And then there was three groups, people listening to trigger sounds. I'm sorry, three conditions, two groups, people with misophonia and without. And there was three conditions. And what they found, sorry, I keep gulping, by the way. So the one condition was trigger sounds. The second condition was just unpleasant sounds. And then the third condition was just neutral sounds, okay? Misophonic distress rating. Then there was just the antisocial rating, based on the control group. And then both groups did the annoyance ratings, misophonics versus controls, okay? But here's what they found, which is very interesting. And this is really cool. This was the first real empirical objective evidence that misophonia is a neurological condition or has underlying neurobiological causes. They found the left anterior insula and the right anterior insula, which I talked about before, the activation for trigger sounds was enormously high for misophonics versus controls, around the same for the unpleasant sounds, which makes sense, and then neutral sounds as well, both with the right anterior and left. Not only that, but look at this third graph. Not only was there activation of the anterior insula, but when we were in the scanner, we were rating how bad on a scale of 1 to 4 was that trigger sound versus 1, 2, and you had to do it with your fingers. I was laying in the scanner 1, 2, 3, 4 for my rating. And they found that there was a correlation between the amount of activation and the amount of intensity for listening to the misophonic sound. And they found that there was some activation for the left anterior insula where the misophonic was listening to the trigger sound, but it was a 1, but a lot more activation when they were listening, the anterior insula was activating when the misophonic sound was worse, like a 4. And both in the right and the left. So that was really cool. That was a really, really cool result. They also found, talking about brain networks again, right? The left anterior insula, they used as what's called a seed region, and they found that there's abnormal functional connectivity, and I forgot to define that term. Let me define it now, actually. Okay, let me define it now. Yeah, let me define it now. Look, so let me show you the brain here. Put a little bit more light on this, on the subject here. Sorry about that. So here's my brain model. Okay, let me try and explain this to you. So here's my brain model. So structural connectivity is when different brain areas are structurally, physically, anatomically connected to one another. Functional connectivity is something different. Functional connectivity is where you put the person in a scanner, right, and you let them listen to the misophonic trigger sounds. And like, for example, this area of the brain back here and this area of the brain over here might not be directly connected to one another. Like, for example, I'm not directly connected to Mark Zuckerberg on Facebook, but there's probably only 3 hops between I know somebody who knows somebody who knows somebody who knows Mark Zuckerberg, and the same with you all as well. That's what's called a small world network. But let's say Mark Zuckerberg is over here and I'm over here, and we're not directly connected to one another. But when we're listening to misophonic trigger sounds, both this area of the brain and this area of the brain, that is me and Mark Zuckerberg, become active at the same time. We're functionally connected, even though we're not directly structurally connected. I hope that made sense. You can ask me questions if not. Okay.
That was
So
an
that's what
excellent description, Michael.
and then
Thank you.
okay, cool. And then there's something called causal connectivity, which is a step beyond that. And that is when this area of the brain causes this area of the brain to become active and not the other way around. That's what I studied in the brain. And so that's what he used here in this study. He looked at directed and undirected functional connectivity. And he found that you see here, misophonics versus controls, trigger sounds versus just unpleasant sounds. There was an abnormal functional connectivity between the anterior insula and what's called the precuneus. Abnormal connectivity between the left anterior insula and the prefrontal cortex here versus misophonics versus controls. Same thing with the, excuse me, sorry, hold on for a second. My apologies. With the hippocampus, and same thing with the amygdala in misophonics versus controls. So he was using functional connectivity analysis to show that there's abnormal functional connectivity between these different areas of the brain, okay? And the last thing that he showed, which was really interesting, is that there was higher level of, if I go back to the neuron here, so this stuff here are called glial cells that wrap around the white matter. That's what makes it white, actually, these cells. And it makes it easier for these electrical signals to travel through. It's like insulation, just like how the wires in your walls work, okay? Same thing about how like the wire works here. It's insulated, right? The brain uses the same thing to allow these signals to travel through, and that's the white matter. And he found that, and the more insulation you have, by the way, the faster the signals travel. This is the problem with multiple sclerosis. This white matter, these cells degenerate and the signals can't get through as they should. He found that, interestingly, here's the study here, that there was extra white matter or extra what's called myelination in the ventromedial prefrontal cortex. That's the area that's connected with the left anterior insula in the misophonics versus controls. Okay, he found that there was extra of that white matter. Oh, I have it here on the slide here. I didn't even need to go back. So there was extra myelination or extra white matter. So the signals might be, that's what's causing this abnormal functional connectivity. One of the things that's causing this abnormal functional connectivity is the signals aren't traveling as they should travel because there's too much myelination. That's a crude description of this, okay? The other study, how am I doing on time, Jen?
I'm going to check that for you and let you know in just one moment.
Okay, so let me just go through this second study. This is the study that replicated or reproduced Dr. Kumar's study. And they found pretty much the same thing. The one difference in this study, they looked at the brains, right, what was becoming active. The one thing, they did a couple things different in this study a little bit, but one of the things that they did in their studies was the subjects weren't only listening to sounds, they were also watching videos. So that's the visual aspect as well, which we all know about misokinesia and the visual triggers. And so they wanted to include that as well. They also had a better questionnaire to make sure that these people really had misophonia. They also looked at anger, not just like whether the trigger sounds were triggering versus unpleasant. They looked at like the actual emotion of anger. And yes, they found that these areas, the same areas of the brain lit up in the salience network of the brain in misophonics versus controls.
Just to let you know, Michael, we're at 8 o'clock, which is when we would
Okay.
start question and answers. However,
Yeah, that's
we
fine.
want
I
to
can,
continue because people can also write in questions and answers.
let me just take this, and then I could take some questions and answers.
Okay.
And then if I want to talk about, I could briefly talk about the treatments. I'll really quickly go through the treatments. Okay, just give me like, give me like another 5 more minutes.
Absolutely. Take all the time you want.
Because they're just hypotheses. We don't even know if the grant went through yet. We'll see. Hopefully it will. And yeah, they found the right insula, the right anterior cingulate cortex, and the superior temporal cortex, the auditory cortex, were all becoming activated in misophonics versus aversive, to just the unpleasant sounds versus the neutral sounds. So they really found the same stuff, but even more. The point is here that they found the same stuff as Kumar, but even more controlled, even more rigorous.